Good afternoon, everyone. It is a great honor for me to participate in Oncomine World 2024, and I'd like to express my gratitude to Thermo Fisher for inviting me to share our experiences and findings on the use of the Oncomine Precision Assay. Here's our disclaimer. So first of all, I would like to highlight that the findings that I'm presenting today are some of the work that we've done during my time at the Japanese Foundation for Cancer Research. I was quite fortunate to be involved in a Japanese national project for the establishment of an AI hospital system. Within that project, we've conducted a series of clinical studies that looks at the use of liquid biopsy for cancer management. So before I move on, I'd just like to share some quick background on circulating tumor DNA, or also commonly known as ctDNA. Liquid biopsy, in particular to ctDNA, has been of great interest to the oncology field in recent years. This ctDNA are tumor-derived fragment DNA that is flowing in the bloodstream. So they're usually around 150 to 200 base pairs long, and there isn't actually that much of an abundance in the blood. So because of the short fragment length as well as the low abundance, it actually makes them very difficult to be accurately and sensitively detected. And that's why a lot of work has been done to try to get that sensitivity level up. Why are they of so much interest is because they actually harbor the genetic alterations that are present in the tumor, and that the type of genetic alterations can range from point mutations to copy number alterations, rearrangements, and also methylation changes as well. They have a relatively short half-life of approximately 30 minutes to 2 hours. And because of this short half-life, it actually gives us a real-time reflection of the tumor. And this then can actually be done through a minimally invasive procedure of blood draw. So all these unique features of ctDNA mean that it allows us to perform repeated sampling so that we can actually characterize and capture the temporal heterogeneity that develops over time, which is a capability that's often not feasible when using tumor biopsy. So these favorable features have prompted the researchers to explore the potential clinical utilities of ctDNA for implementing precision oncology. These utilities included the use of ctDNA for early diagnosis and screening, also to see how ctDNA can be used for detecting minimal residue disease after surgery and how that may predict prognosis. Also looking at how it can be used for therapy selection, usually through the genomic profiling of cell-free DNA. And lastly, to explore the use of ctDNA for disease and therapy monitoring. So really to see whether we can capture and detect recurrence earlier than our typical radiological method or our tumor serum markers as well. So there is an increase in the utilization of liquid biopsy assays for genomic profiling in recent years, and this is largely supported by the approval from both the FDA and PMDA. PMDA is pretty much the Japanese equivalent of FDA. And in addition to that, there are also a large number of studies have shown that liquid biopsy-based assays do allow a much shorter turnaround time for reporting results compared to our traditional tissue-based assays. But despite all these advantages, cell-free DNA-based assays remain largely as a complement to tissue-based assays. So the first thing that we did was we tried to understand what are some of the hurdles and barriers that are actually limiting the use of liquid biopsy in the clinic. So on this slide, I've summarized some of the limitations of cell-free DNA-based assays. And what we realized is that these limitations can be broadly grouped into two categories. The first trip is that the overall procedure is largely non-standardized. And what I mean by that, it actually starts from blood collection through to cell-free DNA extraction, the idea of library preparation, sequencing, and even the report template. So because they are non-standardized, clinicians often find it difficult to interpret the results, and this could be also due to the potential false positives and false negatives that are associated with cell-free DNA assays. So that's one group. But on the other end of the spectrum, it's the idea that most of the available assays are actually largely centralized. So that means you will require special storage and transportation of the blood samples, which would bump up the cost, and at the same time, it could potentially reduce access to the patients as well. And so what we're interested in is to evaluate whether it is possible for us to have a decentralized system that could actually allow the whole comprehensive analysis to be done at the hospital or at the research institution, such that it could improve the access and therefore further enhancing the turnaround time as well. And if such system actually exists, then what will be its feasibility? And we wanted to evaluate this by mainly looking at the ctDNA detection sensitivity. So we then came across with this EON-Torrent GeneXus integrated sequencer, and we wanted to evaluate it and see whether it actually addresses all those clinical questions. So this GeneXus integrated system is an all-in-one machine that automates the entire process, starting from cell-free DNA input through to lab reconstruction, and then through to sequencing, and also finally data analysis and possibly report generation as well. So all this, the entire process is actually automated, and it can actually be done within 24 hours. So this automation means that you really need very little technical sort of supervision, and it means that the system can be decentralized and situated at the institution as well. So we evaluated this system or this sequencer with the use of the Oncomine Precision Assay, which was designed to detect targetable alteration through the detection of SNVs, indels, CNVs, and fusion across 50 genes. The coverage is around 40,000x with a unique molecular coverage at around 2,500x with as low as an input of 10 nanogram only, and the limit of detection for the SNVs and indels are approximately 0.2%. So we first evaluated the analytical performance of the assay using a range of cell-free DNA standard controls. Overall, what we've observed is that OPA showed a high consistency in detecting SNVs and indels down to a variant allele frequency of 0.2%. At the same time, it also showed high sensitivity in detecting a range of fusions as well. And because of this analytical performance, we then expanded our study and evaluated the OPA with the GeneXus system using 298 plasma samples that were across different cancer types. So we compared these results with the tissue samples that were sequenced by a different targeted assay. The first thing that we noticed was this high sequencing success rate. So first of all, sufficient cell-free DNA was extracted from all 298 plasma samples with a median sequencing input of 26 nanogram. So we've noticed that the OPA showed a significantly higher sequencing success rate at 99% of samples passing the sequencing QC matrix compared to a success rate of 96% using tumor tissues. We also try to evaluate the failed tissue cases to understand why they didn't pass the QC matrix, and most of them failed due to the insufficiency of tumor content or poor DNA quality of the FFPE tissues. We also found that the sequencing success rate of the tumor tissues was actually related to the age of tissue, where the failure rate actually increases to 10% for any tissue specimens that were collected more than 3 years ago. Then we looked at the ctDNA detection rate, and we found that overall 50% of the samples that we've sequenced detected with at least one ctDNA alteration. And this ctDNA detection rate was highest in colorectal and breast cancer at around 65 to 70%. And it was lowest observed in head and neck as well as in thyroid cancer samples, and that's around 20 to 30%. But overall, this sort of detection rate was actually aligning with the existing literature. We've also compared the genomic landscape detected from ctDNA of the 4 most common cancer types in this sample cohort with the TCGA data that were based on tumor tissues. So as you can see from the diagram, overall it did show a high concordance level except for 2 observations. So for one, we found that the detection rate for TP53 and KRAS mutation were lower for pancreatic cancer. And on the other hand, we detected a higher level of ESR1 mutation and KRAS mutation in the breast and the GI sample cohort respectively. And this is likely to be reflecting therapy-related resistant mutations of these genes. We've also evaluated the detection of clinically relevant alterations. So overall, 29% of the samples harbor at least one clinically important alterations from plasma cell-free DNA, and we define this as level 1 to 4, as well as level R1 on OncoKB. So in particular, we found that breast, GI, and lung cancer cohort showed the highest detection rate of targetable alterations. For the last part of the presentation, I would like to show some of our concordance analysis between plasma cell-free DNA using OPA and tissue sequencing. So overall, we found that up to 72% of our sample showed concordant results. And we defined this by having at least one concordant mutation detected from both the tissues and the plasma, or there were no mutations that were detected from neither of the assays. What we also noticed is that the concordance level was actually quite variant type specific. So we found that around 63% of the SNVs or indels were concordantly detected between the plasma and the tissues, compared to only 12.5% of the CNVs that were concordantly detected between the 2 specimen type. So we wanted to understand what are some of the reasons for this. And what we found was that the variant allele frequency of the alterations that were concordantly detected between tissue and plasma were actually significantly higher than the variant allele frequency of the alterations that were detected from the tissue only. What that actually means is that for alterations that were detected with a clonality of more than 30% from the tissue, the concordance level was actually quite high, so you're looking at around 80%. Compared to those alterations that were detected at clonality of less than 30%, those when the concordance level was only 40%. So what this is telling us is that the mutant clone size itself would affect the ctDNA detection sensitivity. Well, for the CNVs, we've observed something similar. So where you would actually need a much higher copy number amplification. So you're looking at a copy number of 10 or above, or alternatively, you will need a plasma sample that show a very high level of ctDNA fraction, so close to 40%. In another word, in terms of the plasma cell-free DNA, you will need at least 40% of them to be tumor-derived in order for the CNVs to be detected from the plasma cell-free DNA. But this is actually not new, so this phenomenon has also been observed in several previous studies as well. So in summary, Oncomine Precision Assay, accompanied with the use of Eon Torrent Genex's integrated sequencer, showed a high sequencing success rate of 99%, with a low cell-free DNA input of approximately 10 nanogram and a fast turnaround time of 1 day. Sequencing success rate was higher using cell-free DNA than tumor tissues. The analytical performance was that the LOD is approximately 0.2% for the small variants, and 50% of the samples were detected with at least one ctDNA mutation. Lastly, 72% of our sequence samples showed concordant results with the tissue samples. And finally, I would like to thank to all our team members from the Cancer Precision Medicine Center, which was part of the Japanese Foundation for Cancer Research. So our CPM Center was led by Professor Nakamura, as well as Dr. Amanda Lowe, and also our collaborating clinicians from the Cancer Institute Hospital of JFCR, in particular to the Department of Genomic Medicine, who was actually led by Dr. Takahashi. Thank you very much, and I'm open for any questions.